Chapter 4, Biology of the Cell, Part 1. So, cell theory. Cell theory comprises of three main points. The first is that the cell is the basic unit of life. So no matter how tiny you get, even down to itty-bitty little bacteria, they are all still consisting of at least one cell. And that feeds into number two, all living things are made up of one or more cells. So if you don't have a cell, you are not a living thing. So that is why viruses are not considered living. And then the third is that all cells arise from existing cells. There's no spontaneous generation. Human cells come from other human cells. Bacterial cells come from other bacterial cells, and so on. So the study of cells is called cytology. Cytology. And when we're talking about cells, we're talking about things measured in micrometers. So we did talk about millimeters in virtual labs in Unit 1. Well, this is 1,000 times smaller than a millimeter. Light microscopes, most commonly used. They have light pass through the specimen that you see on a tiny little slide. And then there are lenses that will focus and magnify what you see. And in order to see the various structures of import in cells, these samples are usually dyed in some manner to make it easier to differentiate different cellular structures. Nowadays, though, super modern technology, we have electron microscopes. Electron microscopes use a beam of electrons, and they have a massively greater magnification and resolution, so they can see extremely tiny structures. So for instance, here over in this slide, this is coming from a light microscope. We're looking at a tissue sample. Here's some open space. Here's cells forming some tissue with a little hair there is the cilia of those cells. If we use a transmission electron microscope, we can see the cilia up close, actually look inside of the cilia to see the protein fibers that make up the cilia. And there's the plasma membrane. And then with a scanning electron microscope, we actually can see the surface of the cells. So we see all the cilia looking very much like little hair-like or thread-like structures. So we usually think of things in terms of meters and millimeters, but cells are all the way down to 100 micrometers, all the way down to just 1 micrometer for some of these structures found inside of cells, so extremely tiny. We can see a lot of this with the light microscopes, and then if you want to go even smaller than that, you're going to have to get those electron microscopes. Here's a model of a cell. It's a cartoon model. It has a lot of inaccuracies. The colors are off. Again, cells aren't really that colorful. The relative sizes of various structures aren't really correct. You'll never see microvilli, cilia, and flagellum all on one cell. A cell will either have none of these or one. So either it'll have no extra extensions from its plasma membrane, or it'll have one of these. When we talk about cells, we break them down into three main component portions. There's the plasma membrane, which is the structure that forms the border, the surface of the cell, which includes these extensions, flagellum, cilia, and microvilli. It includes the nucleus, the largest organelle that houses the DNA, and then everything between the plasma membrane and the nucleus. And that's referred to as the cytoplasm. Cytoplasm is everything between the nucleus and the plasma membrane. And here's just another view of just the plasma membrane being highlighted. So the plasma membrane is composed of a phospholipid bilayer. That should be familiar. These little spheres with the little tails hanging down, these are phospholipids. When they come together to form the plasma membrane, they form two layers, two sheets. And then they separate the inside from the outside of the cell. So they separate the cytoplasm from the extracellular fluid, sometimes known as interstitial fluid. Close-up of a phospholipid, of course, we have that phosphate group, and we have two long fatty acid chains. The phosphate group is charged, it's hydrophilic, so water-loving, and that's why it faces out, because we are very aqueous. We have lots of watery solutions in our bodies. And then the fatty acid chains, nonpolar, hydrophobic, they face in to each other to try to avoid contact with water. And so phospholipids are the mysterious amphipathic molecules. They are both hydrophobic and hydrophilic. So the plasma membrane's important job is to be selectively permeable. They allow some things to pass through, to cross the plasma membrane with no problems, while other things are blocked and cannot go through the plasma membrane. Selective permeability. Some things can pass through: water, small things like oxygen and carbon dioxide, other small nonpolar substances. Some things cannot. Charged substances like sodium ions, large substances, and polar things. Yes, I know water is polar, but it manages to squeeze through. There are a lot of other things besides phospholipids in the plasma membrane. We have little bitty cholesterol molecules found embedded in with the fatty acids. Cholesterol helps to make the plasma membrane a little more rigid. We have glycolipids. These are fatty acid chains attached to a glucose portion that faces on the outside. We have glycoproteins. Glycoproteins also with a carbohydrate portion facing on the outside. We have proteins embedded in the plasma membrane, referred to as integral proteins. We have proteins that just lie on the surface of the plasma membrane, referred to as peripheral proteins. Proteins, glycoproteins, lipids like cholesterol, glycolipids. The plasma membrane has a variety of functions. And so do the proteins associated with it. So for instance, we have the transport proteins. Transport proteins are integral proteins, go all the way through the plasma membrane, sometimes called transmembrane proteins. They can help move substances across the plasma membrane. They can act as channels, carriers, pumps, basically regulating, controlling the movement of things across the plasma membrane that cannot normally move across the plasma membrane. We have proteins that act as receptors, so they'll bind to a ligand molecule, some chemical produced by some other cell, usually, and they will bind to it, and it'll lead to some sort of effect, some response inside the cell. Examples of ligands include neurotransmitters and hormones. We have identity markers. It turns out those glycoproteins, the carbohydrate portion of those glycoproteins, are identity markers. They communicate to other cells to say, hey, I belong here. Don't destroy me. So they keep the immune response from destroying healthy, normal cells of the body. Some proteins associated with the plasma membrane are enzymes. So they catalyze specific chemical reactions. Substrate comes in, product goes out. Some proteins associated with the plasma membrane are anchor sites. So they secure the cytoskeleton, so these long protein fibers attach to the anchor sites, attaching to the plasma membrane. And then also cell adhesion proteins, again, found on the plasma membrane, but in this case, they are interacting with the adhesion proteins on neighboring cells. So cells from neighbors to each other are able to make protein contacts to adhere, to attach to each other. Important when we talk about tissues. All right, moving on to transport of materials across the plasma membrane. Well, the first big category of transport is passive transport. Passive transport means things are moving along either the concentration gradient, going from high concentration to low, or along the electrical gradient, going from an area that is one charge to an area that is the opposite charge. And the cool thing is this does not require energy from the cell. This is free movement. And everything that is free is better. So here we have a dye dropper. We dropped a bit of dye into a beaker of water. We have a high concentration of dye in the droplet, low concentration in the water. Over time, the dye moves from the high concentration droplet out into the low concentration of the water, and eventually it becomes thoroughly mixed with the water. So that is diffusion. For cells, we have simple diffusion. Simple diffusion is anything that can move across the membrane on its own. So this would be small things, nonpolar things, so oxygen, carbon dioxide, some fatty acids, some hormones, urea. They're small enough, they're nonpolar enough to slide through the plasma membrane. And the movement can go in either direction. They can move into or out of the cell. So when we talk about movement, we're really referring to the net movement. And that net movement is going to go from high concentration to low concentration. So sure, there's a few things that go the opposite direction, but the majority of movement is going from high concentration to low concentration. We have facilitated diffusion. Facilitated diffusion requires proteins, integral proteins associated with the plasma membrane. These are the ones that have opening spaces to allow materials to cross the plasma membrane. They cannot normally move across the plasma membrane. These are for small charge things, for polar things. So for instance, they will allow things to move freely, like sodium ions can go through sodium ion channels freely, constantly entering, or potassium exiting. Or they can be carrier mediated, so they'll allow something to enter, but it is a controlled process only going in one direction. So channels could go in either direction. Carrier mediated diffusion only in one direction and no other direction. And the more proteins you have to allow the transfer, the faster it occurs. Then we have osmosis. Osmosis occurs through the plasma membrane, allowing water to go from high concentrations of water to low concentrations of water. Or the water can pass through special integral proteins called aquaporins. Aquaporins form a channel to allow water to move into or out of the cell, going from high concentration to low concentration. Osmosis could be simple diffusion or it could be facilitated diffusion. But it's always dealing with water. And other things, if they don't have special channels, can't move through the plasma membrane. Proteins can't do it. Glucose can't unless they have a special protein to help them, etc. All right, osmotic pressure. Osmotic pressure is the force, the pressure exerted as water is moving across a semi-permeable membrane, like a plasma membrane. And basically, the steeper the gradient, the steeper the difference between the two water concentration on either side of the membrane, the greater the osmotic pressure. So A here, we have 10% glucose solution versus a 2% glucose solution. Water will then go from the 2% into the 10%. Over here, we have glucose 30% solution versus a 2% solution. This is a much steeper gradient, a larger difference. More water is going to move faster from the 2% side to the 30% side, so it'll have the greater osmotic pressure. So again, 2% glucose means 98% water, 10% glucose means 90% water. So it's going from high concentration to low concentration. And over time, it will reach equilibrium where the water concentration on each side is about the same. Hydrostatic pressure occurs after equilibrium is reached. Hydrostatic pressure is the pressure exerted on the walls of the container by the fluid. So blood pressure is the pressure of blood on the walls of the blood vessels themselves. That's why we don't want it too high, because that could then damage the blood vessels. Cells can gain or lose water by osmosis. It depends on the kind of solution they're in. So the tonicity, the amount of solutes in the solution the cells find themselves in. So for instance, isotonic solutions, isotonic means no net movement of water. So the overall concentration of water outside the cell is the same as inside of the cell. Water enters, water exits, no net gain or loss. Hypotonic, the solution the cell is in is much more dilute than the solution inside the cell. Hypotonic means lots of water outside of the cell, so the water is going to go into the cell. It's going to cause the cell to swell and eventually lyse or burst, which is cell death. And then hypertonic. Hypertonic, the solution outside is much concentrated, more concentrated than inside the cell, so water will leave the cell, enter the surrounding solution. This will cause the cells to crenate or shrivel and eventually cause the cells to die. So cells want to be in an isotonic solution. So glucose, 10% glucose solution for the cell. Outside solution is 5% glucose solution. Solution in the beaker in the cell is hypertonic. The water will move from into the hypertonic solution, so it'll move into the cell. So go from 95% to 90%. Water always goes to the hypertonic solution. That is it for this lecture.